Hello class 9th, hope you all are good. Let's start a new chapter, chapter number 8, Economic Importance of Bacteria and Fungi. In this chapter, whatever is underlined with red pen and whatever I'm explaining, only that part you learn, right? Now, first is what are bacteria? Bacteria, actually, these are the most primitive, simplest unicellular and the smallest microscopic organism. Abel Leeuwenhoek first of all observed bacteria in 1676. Now next is your economic importance of bacteria. All of you know that in general the people think that bacteria, they are harmful and they are disease causing organism. But bacteria are very intimately connected with our lives, both in useful as well as harmful ways. Now, first we will talk about the useful activities of bacteria and bacteria in medicine. How the bacteria help in production of medicine. And first is your what? Antibiotics. Antibiotics are actually the substance which we get from one microorganism, and which is used to check the growth of other microorganism, right? These are called antibiotics. So what are certain filamentous bacteria, that is your actinomycetes, they secrete some organic substances, and these substances check the growth of many other microorganisms, and these are called what? Antibiotics. And some of the wonder antibiotics and bacteria and the bacteria producing them are, first is your streptomycin, which is produced by Streptomyces graeus, and second is your terramycin, and it is produced by Streptomyces ramosus. Now vaccines, next is your vaccines. The heat-killed diluted suspension of pathogenic bacteria of a specific disease is called vaccine. Pathogenic bacteria means the disease-causing bacteria, right? These are actually, vaccines contain dead bacteria or weak bacteria or toxins in a diluted suspension. When they are injected into the body of a healthy person, these vaccines make the body resistant to the particular disease, and the body gets stimulated to produce antitoxins or antibodies, which provide immunity against any future attack of the particular disease. Once a person get a particular vaccine for a particular disease, then for a lifelong he is protected from that disease, right? Now there are main two vaccines, that is your Bcg vaccine, which is produced from living weakened bacteria, and it is used for tuberculosis, right, against tuberculosis. And second is your TAP vaccine, which is used against typhoid, and this TAP vaccine is made from killed bacteria. Okay, next is your serum. Serum is actually a blood plasma which doesn't contain any type of blood cells, means it doesn't contain WBCs, RBCs and blood platelets. And how it is prepared? It is prepared from the blood of certain mammals such as horse, monkey, etc. Actually, first in their body, some disease-causing bacteria are introduced, right? When these bacteria goes inside the body of these mammals, then their body produce antibodies against to fight with these disease-causing bacteria, right? And then after some time, what's happened? Their blood contain these antibodies. Then this blood is taken out from their body, and then from that blood, this blood plasma is separated out, and which is known as serum. So serum containing particular antibodies or antitoxins, which are injected into the body of a patient, and it helps to cure a particular disease. Right. Now next is your useful importance of bacteria in agriculture. Now first is your what nitrification. What is nitrification? It is a conversion of ammonia and other ammonium compounds into nitrates and nitrites and nitrates. That is what is this is called what nitrification. And some of the nitrification bacteria like what nitrosomonas and nitrobacter, they help in this process of conversion of ammonia and ammonium compounds into nitrites and nitrates. Now next is your nitrogen fixation. Conversion of free atmospheric nitrogen into nitrates is called nitrogen fixation, and it is done by some either free living aerobic soil bacteria like azotobacter or some rhizobium bacteria which lives in the root nodules of the leguminous plants. What are leguminous plants? These are pulses plants. The roots of pulses plants, they have these beads like structures which are called nodules. In these nodules these bacteria like rhizobium, they live there, right? And then what they do, they convert atmospheric free nitrogen into nitrates and nitrites, the nitrogen compounds, and which is utilized by the plant for their growth and development. Okay, now next is your nitrogen cycle. Nitrogen cycle is actually nitrogen cycle consists of 5 steps: nitrogen fixation, nitrogen assimilation, decay, ammonification and nitrification. Then is your denitrification and sedimentation. Children, just look at this diagram. I'll just try to explain this one. First step is your nitrogen fixation. Actually, what's happened? Some bacteria, they convert this atmospheric nitrogen into nitrates, right? And this is called what? Nitrogen fixation. Then is your what's happened? These nitrates are utilized by the plants, and they they absorb these nitrates, and then they have these nitrates utilized by the plants in the formation of, they convert them into amino acids. Then these amino acids are converted into into proteins, enzymes and other nucleic acids and etc. Right? Means plants, they utilize this nitrate, and it is called what? Nitrogen assimilation. And nitrogen assimilation is simply conversion of these inorganic nitrogen into organic nitrogen, just like amino acids. And then amino acids are again then utilized in the formation of proteins and enzymes, chlorophyll and nucleic acid, etc. Right? Then these plants are eaten by the animals. So these proteins also goes in the body of animals, means the nitrogen compounds again goes to the body of animals also. Now after the death of these plants and animals, what's happened? Their body decay, and the decomposition of these dead bodies of plants and animals takes place by some bacteria, and they convert these amino acids into ammonia and ammonium salts by the process called decay ammonification. By some bacteria, right? Then what's happened now? Next step is your nitrification. Then again this ammonia or ammonium salts, they are decomposed or are converted into what? Nitrites by some bacteria, and these nitrites are then again converted into nitrates, and this process also called nitrification by some bacteria, right? And these nitrates are then absorbed by the plants, right? But there are some now next is your denitrification. There are some harmful bacteria are also present in the soil which decompose these nitrites and nitrates into free nitrogen, and that's why what's happened, they decrease the fertility of the soil, and nitrogen again what's happened, go back to the atmosphere, right? Now next process is your sedimentation. Now what's happened in sedimentation? Sedimentation is actually the nitrates which are present in the soil, they sometimes washed away to the sea, or they leached deep into the earth along with the percolating water, right? And these nitrates thus lost from the soil surface and are locked up in the rocks, and this is called what? Sedimentation. This process is called sedimentation of nitrogen. So now this nitrogen is what? Assimilated, or you can say it is converted into rocks. Now next is your uses of bacteria in industry. Now first, the bacteria are used in curing of tea and tobacco leaves. Bacteria are also employed for the partial fermentation of tea and tobacco leaves to remove their bitterness and give them a specific flavor or taste, right? And next is your what? That is your tanning of hides or leather, removal of fats, hair and other tissues from the hides of animals. Hides means actually khal jo hoti hai, right? Is also achieved through fermentation by bacteria.
Thank you, children.